Big behavior challenges and opportunities in a new era of deep behavior profiling. The assessment of rodent behavior forms a cornerstone of preclinical assessment in neuroscience research. Nonetheless, the true and almost limitless potential of behavioral analysis has been inaccessible to scientists until very recently. Now, in the age of machine vision and deep learning, it is possible to extract and quantify almost infinite numbers of behavioral variables to break behaviors down into subcategories and even into small behavioral units, syllables, or motives. However, the rapidly growing field of behavioral neuroethology is experiencing birthing pains. The community has not yet consolidated its methods, and new algorithms transfer poorly between labs. Benchmarking experiments as well as the large, well annotated behavior datasets required are missing. Meanwhile, big data problems have started arising, and we currently lack platforms for sharing large datasets akin to sequencing repositories in genomics. Additionally, the average behavioral research lab doesn't have access to the latest tools to extract and analyze behavior, as their implementation requires advanced computational skills. Even so, the field is brimming with excitement and boundless opportunity. This review aims to highlight the potential of recent developments in the field of behavioral analysis while trying to guide a consensus on practical issues concerning data collection and data sharing. Introduction: Measuring Behavior, the Past. In preclinical research, the analysis of rodent behavior is necessary to evaluate normal behavior processes such as memory formation as well as disease states like neurodegenerative diseases or affective disorders. For centuries, the study of animal behavior was limited by the ability of humans to visually identify, record, and interpret relevant behavioral changes in real time. Over the last few decades, first computerization and then commercial platforms have stepped in to automate certain measurements, mostly by providing accurate tracking of an animal's path of motion or nose point, or by counting mechanical events. This has been a game changer for highly constrained experimental testing setups, where a clear behavioral outcome is either expected or acquired over time. Examples include various operant conditioning setups and more complex home cage monitoring systems that can continually measure predefined daily activities. These measures can accumulate large amounts of data over days of continuous recording, yet they do not pose major challenges in terms of data and analysis and will not be discussed further in this review. In contrast to these well automated tests, some of the most popular laboratory tests require ecological assessments, often involving laborious and subjective human labeling. Prime examples are tests that assess the emotional state of an animal, social interaction tests, or tests of prey pursuit. Amongst these tests, assays of emotionality are usually more tractable because a single animal is recorded and analyzed. The open field test is an illustrative example in which the animal is placed in a circular or square enclosure. Originally, only defecation was recorded as a readout of emotionality and sympathetic nervous system activation. Ambulation was soon added, yet early measurements of ambulation relied on manually counting the number of entries into subdivisions of a great flow. The importance of recording subtle, ecologically relevant behaviors like rearing, grooming, sniffing, or teeth grinding was recognized early on, yet the requirement to manually score these behaviors limited the number of parameters that were reported. The advent of computerized tracking through beam grids or video recordings provided automated measures of distance and time spent as different zones of open field. This made the open field test one of the most widely used tests in behavior laboratories around the world, providing a quick readout of both locomotor activity and anxiety. While the number of studies using the open field test has exploded over the last few decades, the measurement of ecological behaviors faded into the background. In today's behavioral neuroscience laboratories, ecological measurements measures as the open field test are largely restricted to scoring a few well-characterized behaviors, ignoring the rich behavioral repertoire the animal displays during the 10-minute recording session. While the automation has thus arguably hurt ecological aspects of behavior, it appears that the advent of machine vision and deep learning tools might now tip the scale in favor of ecology. The future over the last 2 years, we have witnessed a post-tracking revolution. Software packages based on deep learning neural networks allow markerless tracking of multiple hand-picked body points with astonishing performance. From a data perspective, a mouse is no longer only a body-centered nose but has become a complex three-dimensional skeleton composed of moving vectors. This technology has now been merged with supervised machine learning tools which enable the detection and quantification of complex ecologically relevant behavioral sequences with ease and human-like accuracy. Additionally, unsupervised machine learning approaches begin to reveal the stunning repertoire of subtle behavioral sequences often undetectable by humans that can be recorded with robotic precision and simple testing arenas and in various species ranging from rodents to insects and nematodes. This has spawned the field of computational neuroecology, and several excellent reviews have revived the idea to generate a comprehensive behavioral personality profile and ethome or behaviome in analogy to the genome. We direct the interested reader to these reviews, which lay out a thorough general framework for the hopes and challenges big data brings to behavior analysis, and summarize the progress made by recent attempts to leverage unsupervised and supervised computational approaches to map the behavioral space of animals. In the present review, we evaluate the various approaches for recognizing and quantifying ecological behaviors using machine learning and consider the impact of accurate cause estimation algorithms on behavior recognition and quantification. We focus specifically on rodent emotionality, which poses difficult yet tractable challenges for behavioral neuroscience and preclinical work. The findings, limitations, and possible solutions presented here will have important implications for even more ambitious endeavors such as deconstructing social interactions or analyzing predator prey dynamics. Based on lessons learned from computational neuroecology over the last few years, we propose practical solutions for how data could be collected, analyzed, and shared. These suggestions shall aid the establishment of behaviomics as a field with defined standards and guidelines akin to genomics, from human annotation to machine learning. In the early 2000s, commercial platforms specialized in rodent tracking started offering software packages that enable automated quantification of some ecological behaviors such as grooming or rearing in the open field or hand dipping in the evaluated plus males. Embarrassingly, the current consensus is that these commercial systems still perform poorly when quantifying ecological measures in comparison to human results. Thus, human scoring has remained the gold standard for a large set of standard laboratory behavior tests, which is extremely laborious and scales for the number of behaviors scored. Annotating every frame in a 1-hour video with high confidence was estimated to take 22% hours. A study in which experienced raters annotated 13 distinct behaviors reported a time investment of 1 hour per 5 minutes of annotated video. And in our hands, annotating a few behaviors in a 10-minute video takes roughly 1 hour. In addition, several other human factors limit the reliability and reproducibility of human scoring. As a consequence, the vast majority of studies using the open fields test report only automated measures like distance moved and time spent in the center. Even behaviors that are highly informative about the animal's emotional state, like unsupported rearing, have been recorded by only a handful of studies over the past several decades. Today, supervised machine learning offers a sensitive and reliable solution for automatically tracking complex rodent behaviors. Supervised machine learning, the basics include supervised machine learning requires two preliminary steps. In the first step, video or image data is used to generate a number of features that are used as input for the classifiers. Most commonly, animal tracking data from commercial software packages is used to calculate features such as animal length, animal orientation, animal speed, or distance to feeder. These features are usually manually defined and computed with handcrafted algorithms, often based on the point tracking data from the nose and tail base. In this case, animal length is defined by the distance between nose and tail base, animal orientation by the relative angle of corresponding vector, animal speed by the change of position of body center over successive frames, and distance to the feeder by the distance between nose point and the feeder. And the second step, video or image data is manually annotated by human readers to assign labels to individual behaviors. This process is critical as it establishes the ground truth of what the animal is doing at any given moment in time. In the subsequent machine learning step, a mathematical model is then trained to recognize the manually annotated classifications. It does this. The cross-validation data set is part of the manually annotated data that was not used for training. In the cross-validation process, the overall accuracy of the classifier is analyzed when compared to a human annotator. It is important to establish if a classifier only memorizes the initial data or if it manages to generalize. There are multiple methods to improve generalization. The most frequently used ones are regularization and dropout. Finally, confusion matrices are often used to visualize pairwise errors of an analyzed behaviors. The confusion matrix not only contains the accuracy of each behavior but also displays which behaviors are often mistaken for one another between raters. This is the diagram graphical representation. Different supervised approaches. First attempts to use supervised machine learning for automated behavior recognition were made 2 decades ago using single frames from a side view video recording in combination with feed-forward neural networks. From these single frames, 3 points were set and used to calculate features such as distances between points and angle between vectors. However, no regularization or dropout was used, and the data was split randomly into training and cross-validation sets. Consequently, the resulting algorithms performed extremely well on the training set, whereas the cross-validation accuracy dropped to 76%. When different videos were used for both sets, the accuracies were further reduced to 84% and 64% respectively. Later studies started to incorporate temporal information into the feature space and appropriate machine learning methods to improve generalization. A 2010 study used side view video recording of mice in their home cage and computed positional features, velocity-based features, and also motion-based features that encapsulate temporal information. They trained their classifier to recognize 8 different behaviors using an extensive training set comprising 10.6 hours of annotated recording. For classification, they used a model based on support vector machines, which return the most likely behavior at any time, in combination with a hidden Markov model, which takes transition probabilities between behaviors into consideration. They could convincingly show that the human versus model confusion matrix was similar to the human versus human confusion matrix, demonstrating human-like performance. However, there are some notable limitations to this type of automated recognition. First of all, some of the features rely on positional data, which are highly dependent on the setup. Any change in the environmental configuration would render the models unusable. Additionally, the videos were all recorded from the side, so applying it to a test setup with video from a different angle would require the collection and labeling of a massive new data set. A number of other notable studies have emerged over the last 10 years using different types of features and machine learning methods. Most studies calculated features similarly using handcrafted algorithms or filters to describe location, appearance, and movement of the animal. However, the detail and depth of these features varies a lot, with some studies depending on as few as 7 features, while others depend on almost 100. The reported studies indicate that a handful of features can contain enough information to reliably detect some behaviors and that there might be a marginal increase in performance when including more features. However, it also has to be noted that the inclusion of many manually defined features requires extensive development time and increases the chance of including uninformative correlated features that can have a negative impact on model accuracy, implying bigger may not always be better. Tracking multiple animals. In cases where multiple animals are recorded simultaneously, pre-segmentation enables the separation of the two animals. Pre-segmentation uses established algorithms from image processing practices such as the watershed algorithm that can detect borders between objects or animals. Keeping track of the identity of animals is still challenging, especially when animals cross over each other or interact in close proximity, so often different colored animals are used. However, some imaginative methods have demonstrated reliable markless animal identification for tracking groups of similar looking animals, such as zebrafish in setups where animal overlap occurs regularly. These methods depend on purely data-driven approaches where a fingerprint representation of each animal is created in each frame by using a sequence without any animal overlap. In this case, a pixel-pixel pair analysis is used to generate intensity versus distance and contrast versus distance density maps of each animal. After animal crossings, the fingerprint with the best match from the previous sequence is found and the animal ID is updated accordingly. These methods can be employed in parallel to automated behavior recognition to additionally resolve animal identity. Although it is unclear how well such an approach would perform on mice that are not readily identifiable based on fur patterns. Notably, progress in this area is happening fast. As we are writing this review, a new version of the popular free pose estimation software DeepLabCut has just been released, capable of tracking multiple animals including ants, rodents, and primates in simple as well as complex arenas, apparently with astonishing reliability. Given the widespread use of the software package, such an advance will have a major impact, and it is likely that multiple animal tracking will soon be the new standard in the field. New machine learning approaches. In recent years, researchers started to adopt emerging technology from video recognition software, employing 3D convolutional neural networks, which fully pre-filter and encode temporal spatial information on an end-to-end basis. Therefore, no custom algorithms or filters have to be used. To define features, these networks rely on video information directly. Results in recent studies have been mixed. In one report, the algorithm slightly underperformed compared to the original study using the same data, which had used manually selected features. Another study outperformed an older study using the same data. However, this was only the case when heavy data augmentation was applied, and the performance dropped a lot when videos from different setups animals were used. A third study showed a slight increase when using 3D CNN models pre-trained to recognize human behaviors and retained with mouse home cage behavior data from a previous study. However, in this study, only 12 similar side view recordings from the same setup are used. So it cannot be assessed how well it would perform with a different setup. In parallel to these efforts, supervised machine learning based methods have also been developed to automatically recognize mouse social interactions based on ultrasonic vocalization patterns and to recognize emotional states from facial features of mice. It will be exciting to see when all these multi-level analyses converge to construct a multi-sensory behaviome.